The process of meiosis does produce genetic variation. And this is why when we talk about the daughter cells that we get from this process, we do say that they are all genetically different from each other, and then also different from that parent cell that they came from. There's a couple of different ways or steps involved in meiosis that contribute to this variation. So if we talk about a few of those, the first one that I want to discuss is what we refer to as crossing over. And the process of crossing over is going to involve homologous chromosomes. So here would be one homologous chromosome, and then I'm going to draw another one just in a different color so that we can see that they are different from each other. So those are meant to look the same. So if we talk about the pieces that we have here a little bit more, when we're talking about the two sides of the blue one or the two sides or the two chromatids of the red one, these are what we call sister chromatids. So the two red ones are sisters, and the two blue ones are going to be sisters. And the individual sister chromatids, they should be identical to each other because these were copied one from the other. Well, when we talk about a red versus a blue one, these are what we call non-sister chromatids. And the non-sister chromatids, those are very similar to each other, but they're not identical. And that's because they do come from different members of that homologous pair. So they've got the same loci, but just a little bit different. So with crossing over, what this process is going to do is we are going to exchange pieces of DNA between the non-sister chromatids. So to do this, they are actually going to break in exactly the same spot. So when they break in that spot, they are then going to exchange those two pieces with each other. So if I try to draw this on here, what we'll end up with is that we're going to have a difference in the tips here, okay? Because what once was attached to the red one is now on the tip of the blue one, and vice versa. So the end result is that we've got these new chromatids. They have new combinations of the actual versions of the genes. Okay, so new chromatids that we didn't have before, part red and part blue chromatids. This does take place during prophase 1, and that would be during meiosis 1. And this process, it does take quite a bit of time, and for that reason, prophase 1 really is the longest of all of the meiosis phases. And it does take up about 90% of the whole time that's spent in meiosis. And that would be meiosis 1 and meiosis 2. So it's a very time-consuming process, but that genetic variation that we get from that is going to be so important in the long run. The other way that we're going to get some genetic variation is through the process of independent assortment. And so independent assortment is going to apply to what happens during metaphase 1. So if we look at metaphase 1, and this would be of meiosis 1, if we just look at that a little bit closer, during that phase of the meiosis process, we are going to have tetrads that are actually lining up along the center of the cell. So I'm drawing a few tetrads here. The tetrads are where you have the homologous chromosomes actually held together so that we see 4 chromatids together. So that's where the tetra part comes from. So if I just label these, each one of these structures is what we call a tetrad. Those are actually formed during prophase 1. But during metaphase 1, these tetrads are lining up down the center of the cell. Well, what the independent assortment refers to is the fact that each tetrad lines up independently. And that's independent from the rest of the tetrads that happen to be there. And what this means is that they could arrange, such as what we see in this picture, They could arrange so that perhaps we had all the blue ones down the same side and all the red ones down the same side. They can be any number of variations, and this is really just limited by the number of tetrads that we have inside the cell. So independent assortment has to do with each one lining up independently. The way that they line up will affect what we end up with when we do go through anaphase 1 and telophase 1, and ultimately what we end up with when we finish telophase 2 at the very end. Okay, so this is also a very important contributor to the genetic variation that comes from meiosis.